Hello everyone, this is Tejaswini and in this video we will discuss about how to know alpha and beta forms, what is the difference between D and L configuration and Epimers and Anomers, as these two terms are confusing ones, and how Epimers are different from Enantiomers and Diastereomers, what are meso compounds, how meso compounds are different from Enantiomers, and what is the racemic mixture. As in last video I told you what are enantiomers and diastereomers. Enantiomers are not mirror images, but they are not superimposable, and diastereomers are not mirror images but two isomers with same connectivity, same molecular formula but different arrangement of atoms in space. I'll write here the definition of enantiomer. Enantiomers are non-superimposable mirror images. Suppose I draw here one molecule which is glucose, OH, H, OH, OH, H, OH, H, H and CH2OH. As in last video I told you, this one is penultimate carbon atom and this aldehydic carbon atom is anomeric carbon atom. And if hydroxyl group on penultimate carbon atom is at left side, then the structure is D and this molecule is glucose. Therefore, this glucose has D configuration. And for L glucose, this hydroxyl group on penultimate carbon atom should be at left side and remaining molecules are also should be at left side. Otherwise, see here, if I draw here two structures, OH and remaining right side molecules are here as it is, only changed this OH at left side, CH2OH, H, OH and H, CHO. This molecule is not L glucose, this molecule is L iodose. Therefore, I told you, for L configuration of glucose, all the molecules are at right side should be at left side, and this should be the mirror image of D glucose, like this: CHO, OH, H, OH, OH, H, OH, H, H and CH2OH. Now this is L glucose. See here, hydroxyl group on penultimate carbon atom is at left side, therefore L glucose. See, we can say that right-handed and left-handed forms of chiral compound. This one is chiral compound, this one is also chiral compound. I will clear this. You guys understood now? See, right-handed and left-handed forms of chiral compounds are enantiomers. See, they are mirror images of each other and they are non-superimposable mirror images. It means enantiomers can be right-handed and left-handed forms of chiral compounds. Suppose this one is the D glucose, CH2OH, H, 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 OH, H and H. Then L glucose will be mirror image of this. I'll draw here like CH2OH, H, OH, H, OH. See, all are at opposite side. Also, this one is also L glucose. This is L glucose. This is L glucose. This one is also L glucose. Sorry, this is D glucose. And these two are mirror images of D glucose. Therefore, these two are enantiomers as they are mirror images of each other and they are non-superimposable. Now I will tell you how to know whether the structure is alpha or beta, and before going to understand alpha and beta structure, remember one thing: if there is a cis form, then the structure is beta, and if there is a trans form, then structure is alpha. Alpha, remember this. Suppose I draw here D glucose, OH and CH2OH. H, OH, H, OH, H, OH. Suppose this CH2OH on penultimate carbon atom and hydroxyl group or anomeric carbon atom are in trans form, then the structure is alpha and the molecule is alpha D glucose. If this hydroxyl group at upside CH2OH and remaining is same, then this CH2OH and hydroxyl group are in cis form. Therefore, the structure is beta. Remember, see here, this is alpha D glucose and this is beta D glucose, as CH2OH and OH are in trans form and here they are in cis form. Remember one thing: for alpha and beta, we have to consider CH2OH on penultimate carbon atom and hydroxyl group on anomeric carbon atom. From this we can know whether the structure is alpha or beta, anomeric carbon atom. These two structures are known as anomers. Anomers are what? Alpha and beta. Configuration of same molecule is anomers. Suppose this is the molecule with more than one chiral carbon. If I change the configuration at one stereogenic center, suppose if I change here configuration of one stereogenic center, then this molecule is D-mannose, and D-mannose and D-glucose are epimers. Now epimers are what? Diastereomers with more than one chiral carbon, but configuration of only one chiral carbon is different. See here, rest is same, but configuration of only one chiral carbon is different. Now we can draw also one molecule here, CH2OH, CHO, H, OH, OH, H, OH, H, OH. See, in these two structures, rest is same except this configuration at one chiral carbon. Here hydroxyl group is at right side and here hydroxyl group is at left side, and this structure is D-galactose. and these two are epimers. Here I will write what are epimers. Epimers are diastereomers with diastereomers means they are not mirror images of each other, with more than one chiral carbon but configuration of only one chiral carbon is different. Now I will tell you what are the meso compounds. Meso compounds are compound with plane of symmetry and they are optically inactive although it contains two or more chiral carbon. See here, there are chiral carbons. Sorry, not here, this one. And this is plane of symmetry. This cuts the molecule into two equal parts and one part is mirror image of another part. Therefore, this molecule is meso. This is not meso compound as these two are not mirror images of each other. These parts after cutting this molecule, and this is also not meso compound. This one is a meso. And what are the racemic mixture? Racemic mixture is the mixture of two enantiomers with equal proportion of isomers. And in next video we will see next part of carbohydrate series. Keep studying. Thank you.